Hello, I'm talking to you. This is Goddess of Science Biology, and I'm Mrs. Bolden. And today we're going to talk about translation and the central dogma in part 9 of my molecular genetics series. Okay, let's go. So, translation is taking that mRNA that we made during transcription, and now we're going to read that recipe and put together a protein using that recipe. So really, translation in a nutshell is just making protein using the mRNA recipe. Okay. So we have, and back along when we talked about different types of RNA, we talked about tRNA, and tRNA is transfer RNA, and it kind of looks like a little clover leaf here. So, oh my gosh, where's the picture? I haven't found the picture. Anyway, so it kind of looks like a clover leaf, and eventually there'll be a picture here. And a specific amino acid gets attached to one end of it, and then on the other end of the clover leaf, kind of the top leaf in the petal or leaf in the clover leaf, is what we call an anticodon. It has 3 bases in the middle of it that's opposite to the bases in the mRNA. So if the mRNA codon is CAU, the tRNA anticodon is going to be, well, let's see. C always takes G. A in RNA world takes T. And U is going to take the A. So it's going to be G, U, A. Should be our anticodon. Okay, and that's going to be kind of important here, because it binds temporarily to the mRNA codon. So this thing comes in, and because it's mRNA is red, and then the tRNA can fit in there quite nicely, and it holds on to it for a wee bit so that the next piece of tRNA can come in, and the 2 amino acids that are hanging off the tails of those 2 can attach to each other. So this allows only the correct amino acid to be attached. And finally, here's the picture of the tRNA. The amino acid gets attached up here, and these 3 down here, these 3 bases down here, are the anticodon. And as you can see, if the codon is G, then the anticodon opposite that is going to be C. And C is going to be opposite G, and then another C is going to have another G. So only a particular amino acid that is attached to this particular piece of tRNA with this particular anticodon can bind in here the way it's supposed to. So it's really kind of a crucial thing to make sure that the right amino acid is going to be attached to the right spot in the protein. So let's talk a little bit about a ribosome. Ribosome is really 2 pieces that kind of come together. You can kind of think of them as 2, if you have like a hamburger roll, you have the top piece and the bottom piece. As a matter of fact, they kind of look a little bit like that. If there's no mRNA that needs to be worked on, these 2 kind of just float around in the cytoplasm waiting for, or they're attached to the ER waiting to work. So when an mRNA comes along, the 2 subunits kind of make a sandwich out of that mRNA. So the top of the hamburger bun and the bottom of the hamburger bun come together with the mRNA in between them. Then a tRNA with UAC anticodon binds to the UAG codon on the mRNA. Remember, mRNAs always start with AUG because that's the start codon. So the anticodon that would be on the tRNA would have UAC because that's what's opposite. That would be what would fit in with the AUG codon. And on the other end of this tRNA that has the UAC anticodon, it has a methionine, and we know that methionine amino acid starts all proteins. Sometimes it gets snipped off later, sometimes not. So the tRNA with the next anticodon, then the ribosome is going to sit right there, and there's another slot for the next tRNA with the right anticodon to come in there and bind. And then once that happens, the next amino acid, of course, is going to be on this new tRNA that's right next to the one we just placed. And then the amino acids on the tails are going to join together, which is great because this is what we want. We want a whole string of amino acids to be joined together. The tRNA, the first that was in there first, that had the methionine attached to the tail of it, now gets kicked out. Everything, the bun shifts down the mRNA, and it reads the next codon on the mRNA, finds the new tRNA. It attaches in there the two amino acids that were previously connected, now attach a third amino acid to it, and it keeps going down the line, you know, kind of chugging through all of these codons until it gets to a stop codon. When it gets to the stop codon, the two pieces of ribosome then come apart, and the protein is released, which is great. So the release factors cause the mRNA to be let go, and the ribosome comes apart, and the protein is now free to do what it needs to do in the cell. So here's kind of the overview of central dogma, and I apologize for my arrows to already be on the screen. So remember when we started talking about this chapter, we talked about the central dogma, which is an established principle. And it's from DNA makes RNA, which gives us protein. And these are kind of an overview. Since you've heard all of the different pieces of this, now you have the overview that kind of puts it all together. So the DNA, which hangs out in the nucleus, undergoes transcription, and we know that transcription makes mRNA. The mRNA then goes to the ribosome, which is not in the nucleus. The mRNA has to leave the nucleus, find the ribosome, which is either floating around free in the cytoplasm or attached to the endoplasmic reticulum. The ribosome then carries on translation, in which tRNA brings amino acids and attaches them to each other, and it makes a protein. So that little flow chart right there is really the central dogma in a nutshell, just giving you an overview of all the different processes and what the products are and what ultimately comes from it, which is a protein. All right, that was a lot. Thanks for joining me. Have a great day.